Thank you so much, everyone, for being here. I'm Daniela. I'm a PhD student here at the Paris Brain Institute, and it's with great pleasure that I introduce the first session of the day. This session is entitled Emergence of Individuality, and in this session, three incredible speakers, Bassem, Dana, and Daphne, will present their work on how individuality exists and is an important factor for the survival of a species, being it a fruit fly, a mouse, or a human. Each speaker will do a 20 minutes presentation, and at the end we'll have the round table. So during this round table, we'll have time to answer all the questions, but don't be surprised if after each speaker presentation, you don't have the time to ask the questions. Save it for the end. And so without further ado, let me introduce you to the first speaker of our session, Bassem Hassan. Bassem did his PhD in molecular genetics at The Ohio State University in the United States. And after he did his postdoc research on early neurogenesis in both mouse and Drosophila. In 2001, he was recruited to establish the first Drosophila lab in Belgium. And 5 years later, he moved his team to the Paris Brain Institute, where he became the scientific and deputy executive director. His research focused on understanding the genetic mechanisms that regulate early nervous system development using diverse models such as fruit flies, mice, and human cells. And in today's talk, he will discuss how variability in neuronal circuit wiring arises during development and its potential relevance for innate behavioral variability. So please welcome Bassem by a big round of applause.
Thanks, Daniela, for the kind introduction, and thank you, Christiana, for the comments. I can tell you, as a person of color, everything you said about gender diversity, really, especially the sense of belonging, of feeling that you're part of a community where you, at least in the days where I was a student and a postdoc, it was extremely rare to have someone of my background in meetings and conferences and so on. So it takes a lot of perseverance. And I can only imagine what it would be like for women of color, for example. So I think we still have a very long road to cover. Anyway, be that as it may, let's get to the science. So the principal take-home message of the talk today is this. The reason your car or your phone or whatever instrument you're interested, your computer you're interested in using works the way it works is because of the way it was put together, right? And this absolutely the same thing is true for your brain. And from that perspective, if you truly are interested in understanding how your brain functions, then I would contend that we can never understand truly how the brain functions if we don't understand how the brain was put together. Except the brain was, of course, not put together. It's a self-organizing, evolved entity. And so the term we use for that is how the brain developed, right? And I will try to show you this, an example of this through the talk today. Now, I already alluded to it. If you want to understand the principles of how a brain develops, then you must, of course, understand the the fact that the underlying principles are principles that are guided by evolution by natural selection, which today remains truly the only biological theory that has overwhelming evidence for it, and for which appears to contextually explain almost everything we know about biology, and without which we really have no good frame of reference in terms of understanding biological diversity. And of course, as you all know, probably better than I do, that one of the foundational ideas of evolution by natural selection is that all life forms are related and arose at some point from a common ancestor. And what that means is that once evolution figured out, and pardon the anthropomorphism, figured out a way to make a brain, it never went back, which means that all brains that exist today, all brains of all species, are essentially variations on a foundational theme, right? So from that perspective, The details are, of course, very, very different, but the principles are the same. And this is why we can use simple models in science to solve potentially very complex problems. And I'm illustrating this in a very biased way using, standard model organisms that we study in our approaches, the fruit fly, the mouse, and more recently, human neural stem cells that from which you can derive neural-like structures. And so, for the purpose of the talk today, I will tell you about our efforts working in fruit flies, which generally speaking have much, much smaller brains than most humans. I'm not sure that's true for all humans, but than most humans at any rate. And yet, of course, develop and function according to fundamentally the same principles, and from which we can, as I will show you, learn a lot, even surprisingly, about things like individuality. And for lack of a better term, animal personality, essentially. And we can discuss whether that's an appropriate terminology or not later. Now, if you look at the brain of a fruit fly and you trace you all know that brains are made of cells called neurons and glia, and that the neurons are the computational units of a brain, and that they are cabled together in circuits. They're wired in circuits, which then give rise to networks. And if we now trace a few of these wires called axons and dendrites that cable neurons to each other, just a few, in the brain of a fruit fly, this is one of the kinds of the pictures that emerge. And in fact, in looking at these types of diagrams, already in the early 20th century, in the early 1900s, by people like Santiago Ramón y Cajal and others, metaphors of electrical wiring of a brain started to emerge. And we use those metaphors today. We think of circuits as brain circuits like electrical circuits, and many people talk about the brain like a computer. And so this is one of those metaphors in which basically this could be the electrical wiring diagram of a motherboard, an apartment, whatever it is, right? And so what characterizes this particular circuit is the fact that every connection is known. And in fact, if you have this as a blueprint for an electrical circuit, any electrician can build precisely this circuit, and it will function every single time exactly the same way. And not only that, it would be completely predictable how it will work, but it will also be completely predictable what every intervention or dysfunction in this circuit will lead to, right? And so it's very tempting to think of the brain as an electrical wiring circuit, but nothing could actually be further from the truth. It's the absolutely worst metaphor I can think of for a brain, is to think about it as a computer, for example. This is a tiny cubic millimeter, a few microns, cubic microns, of the brain of a fruit fly. This is just a tiny piece of the brain of a tiny brain, right, of a tiny organism. And you see all the cables, the axons and the dendrites, and all the synapses that they form, and so on. Needless to say, this looks absolutely nothing like an electrical wiring diagram, right? And yet from this mess, because there's no better term for this, will emerge amazingly predictable distributions of behavior, incredible adaptation, and amazing things that fruit flies and any other organism can do. I mean, and the evidence for that is, leave a ripe banana in your kitchen, the fruit flies will find it, they will come, and then try to catch or swat one of them and see how good you are at doing that with your huge brain with 16 billion neurons in your cortex. So, in order, therefore, to make sense out of how this mess might give rise to behavior and what explains the fact that behavior is variable across individuals, perhaps it's helpful to start by stating the obvious. And the first thing that is worth stating, assuming the slide went, is that the brain does not exist. So the idea that we're working to understand the brain has some sort of a philosophical, I would argue, quasi-religious connotation to it, this obsession with some ideal that everything that exists in reality, and I blame Plato for this, that everything that exists in reality is some sort of a failed attempt to achieve, this sort of idealized thing called the brain, right? There is no such thing as the brain. What truly exists, unfortunately, I've made the slides go too fast. In reality, and as far as I'm concerned, as scientists, the only thing that we should be interested in is reality. And in reality, there are only individual brains, right? So in fact, the brain that we're trying to understand is not some sort of an ideal thing that real brains are a failed attempt to achieve. It's a kind of a pathetic representation of what reality is. It's not a good thing, it's a bad thing, from my perspective at least. And those of you who know me know that I tend to be unanimous in my perspectives. So. Second obvious thing is that brain development matters for brain function. And of course, you could argue that it does so in a trivial sense. Right, so now let's imagine two parts of your brain, one part made of, I don't know, socialists and conservatives trying to talk to some other part of your brain made of ecologists. And there's supposed to be some sort of a communication between those two parts of the brain. And if that never happens during development for some reason, then it's not very difficult to come up with the idea that whatever signals that first part sends will not be interpreted by the second part, and as a result, some anticipated response will not happen, right? So that's trivial. But in a somewhat less trivial sense, you could imagine that those two parts of your brain are connected in diverse ways in different individuals, right? And the question then is, does that matter? Right? Perhaps the only thing that matters is that these two parts of your brain can connect to each other in any which way, and that there's no so-called evolutionary constraint on the specificity of this connectivity. That's a possibility. But we don't know that that's true, right? And so that's the kind of question that we wanted to tackle with these series of studies. And to make matters even simpler than using a fruit fly brain is we use just one set of neurons in the fruit fly brain in the hope that understanding how that one set of neurons develops and guides behavior might actually tell us something more general about how brains develop and explain behavior. And what's fascinating about this tiny set of neurons called dorsal cluster neurons, but that doesn't matter, and none of the details matter here. You don't have to remember any of this. Is that all of these neurons that you see up here, I don't know if I can point, doesn't matter, on either side, arise all of them from a single mother cell early in embryonic development. And yet, in any given fruit fly, the number of neurons that are born from the single progenitor can vary between 22 and 68. And not only can they vary across individuals, in fact, the number of neurons on the left side and the right side of the same brain is not the same. And generally speaking, the left side and the right side of the same brain have the same genome. Now, furthermore, each of these neurons will send an axon to the other side of the visual system. So what you're looking at here is the visual system of the fruit fly. Again, it doesn't really matter. And the number of axons that will reach this distal target area is going to be variable in a way that does not correlate with the number of neurons, okay? And so on and so forth. So each of these axons will make one of two choices. Either send its axon each of these neurons, will make one of two choices. Either send its axon all the way to the end, or actually stop the axon somewhere earlier in this dense green area that you see called the lobula. Again, the names don't really matter. So this is better shown here, where we can label individual neurons, and you can see there are two categories, the L and the M categories of these neurons, and you can see that the L category, the axon stops early, and the M category, the axon goes all the way to the end. So it's a binary choice. Every neuron is making one out of two possible choices, right? And so Maeva Andreațilavo in the lab wanted to understand the basis of this binary choice. What makes a given neuron make that decision, and why is that number variable between animals and between two sides of the same brain? And so she used live imaging, as you can see here, so we can actually get in there and image the behavior of a single neuron and see what choices it's making during development, right, while the brain is still developing. And when we do that, we learn something super interesting. I'm summarizing about 10 years of work in this single slide that was done by Maeva, but before her by other students in the lab, notably Marion Langen, who was a PhD student at that time. And what Marion initially found, and Maeva confirmed and extended into a whole big model, is that what these neurons are doing is they're competing with each other, so to speak, for the possibility of sending their axon more distally, for further growth, right? And stochastically, one neuron will, quote unquote, win this competition and send the axon all the way to the other, all the way distally. And all the other neurons will react to this and keep their axon proximally, okay? So the identity of a given neuron doesn't matter for its ability to target distally or proximally. What matters is the discussion between a number of neurons about what the optimal output of a conversation rather than a competition. Saying it, I realize what a male term that is to use. A conversation between these neurons for the optimal outcome. And so the number of neurons, their arrangements, their interactions, the noise in their molecular machinery at any given time in development, all of this contributes to whether the decision will be for that neuron to extend distally or proximally. But what also matters is what all the other neighboring neurons are doing, you see? And this is a process that's very well known in biology called mutual inhibition, and it's known to be, and very well demonstrated to be, a stochastic process, okay? And so what Maeva was actually able to show is that there's a series of timed stochastic processes that will result in some probability of any given axon extending either distally or proximally, which means that without watching the process happens, it is impossible to a priori predict the outcome of a developmental process, even if you know the entire genome and the entire transcriptome of every cell, okay? So you have probabilistic processes resulting in probabilistic decisions that in the end give rise to variability between individual brains. Okay? Very, very simple developmental principle allows the emergence of a huge amount of diversity in the wiring pattern of these neurons. So in some sense, we've understood that the genome, okay, a given sequence of a genome encodes a distribution of outcomes. And so in other words, even genetically encoded processes are non-deterministic, which therefore would allow us to conclude that the genome by definition is a probabilistic algorithm. It's not that we cannot predict the outcome in a given individual from a given genetic sequence because we don't have yet enough information about how the genome encodes the biology. It's even if, I would contend that even if we knew the meaning of every single base pair in the genome, the best we will be able to do is to predict a probabilistic distribution of the outcome. And we just have to accept that reality and live with it. But back to the initial question, does this actually matter for the behavior of the individual? So what if this particular circuit is wired differently in each brain? And this is the work of a former postdoc in the lab, Gerrit Linneweber, who has now his own lab in Berlin, and who wanted to ask exactly that question. And so I showed you that the left and the right of the same brain do not develop the same circuit, and of course, different individuals do not, therefore, by definition, develop the same circuit. And so that creates a kind of a distribution of left-right wiring asymmetries in these flies. And so this is, for example, two flies, one called Omar and the other called Fred. The French among you will understand the reference, I hope. And you can see that Omar has a completely asymmetric wiring pattern, whereas Fred has a very symmetric wiring pattern, and the question is, does that matter? And so to address this question, we used an assay called Burden's paradigm, in which these poor fruit flies are put on a platform surrounded by water with their wings clipped so they can't fly away. And they're walking between two absolutely identical visual cues that are just dark stripes on a lit background. Okay? And because flies are attracted to high contrast objects, they will spend their life walking back and forth. And you will see this in this video for a second. So this is going to be the hand of Garrett putting the fly in there, and the fly will start going towards one end, trying to cross the water. It can't. And it'll go to the other side, and it will try to get to the stripe, and it can't. And so it'll think about it for a second, and then it will go to the other side, and then goes back. Maybe this way there is a way. Nope. Oh, okay. Well, what do I do now? So I'll go to the other side. And they will do this for about 15 minutes, and then we stop the experiment. And of course, we can track their behavior with incredible precision. And from this, we can determine a map of their pathway between the two visual cues. And when we do that, what we find consistently is if the brain is asymmetric, the fly will walk pretty much straight between the two visual cues. And if the brain is symmetric, the fly will walk all over the place between the two visual cues. And we can determine a distribution of behaviors that matches the distribution of asymmetries in the brain. So if we watch a fly behave, we can predict what its brain looks like. Or if we had the chance of looking at its brain during development, we can predict with some probability how the fly will behave. Okay? So in fact, this tiny amount of variation that happens during development due to stochastic mechanisms results in diversity of individual behavior. And if we hadn't understood the developmental principles of why and how the brain develops, we would not have made meaningful predictions about how a circuit might encode variation in behavior. And so this summarizes what I just told you. During brain development, there are stochastic mechanisms that give rise to distributions of symmetries and asymmetries that will give rise then to distributions of innate visual behavior. So I'll stop here. I want to thank the people over the years who contribute to this particular project in the lab in the past and the present. Their names are here. My friend and colleague Robin Hiesinger in Berlin, with whom we've been collaborating for a very long time, Max von Kleist in Berlin, who helps us with modeling developmental events, and other colleagues in Zurich for discovering that similar events can also occur in the development of the vertebrate nervous system. So thank all the funding over the years, the Institute for hosting the lab, and leave you with this little piece of a poem from William Blake in 1794. Am I not a fly like thee, or art thou not a person like me? Thank you very much for your attention.